I've come to the last speaker of the session, and this is Scott Fraser. So I like to watch the World Cup when it comes around from time to time. And back in 2006 is when I really started to understand what a true special athlete someone was when they could make things happen on the field that weren't possible. And so the person, I'm not relating all aspects of Zinedine Zidane to you, Dr. Fraser, but Zinedine Zidane was an amazing French player whose career, he retired in 2006, but he had an amazing ability to not only move the ball himself and to score, but he could also just make other players on the field exceptional. And so that's the sort of player that Scott Fraser is, and he is now currently a provost professor at USC. In other words, the higher-ups in the university recruited him specifically from Caltech to USC to do what he does, and he's the director of science initiatives. He actually came to Southern California from Johns Hopkins University many decades ago. He spent quite a lot of time here at UCI, and putting all of us younger faculty to shame, he was a chairman of his department at 32. I guess putting all of us to shame, he's the chairman of the faculty at his department at 32. But now he too tries to use chemistry, engineering, and physics to try and understand biology in its undisturbed state. In other words, without disturbing or destroying samples, in order to try and understand what's happening at the cellular and subcellular levels. Again, a lot of the work that I did at USC is with Cosimo Arnesano and Scott Fraser, and hats off to you for everything you've contributed, and thanks for coming and talking to us.
Well, thank you for having me. We really thrive on the fun of having people bring us problems that are impossible and then trying to make them possible. So what's really fun for us is building up technologies based on the problems people have. My day job has been to try to develop instruments that let us look at experimental animals and see multiple labels inside of them, either vital labels or in situ hybridization in fixed animals. And what we've been trying to do now is to translate as many of those as we can over to clinical utility, some of them that are starting to work. I need to start by thanking some very capable people. In addition to stealing techniques from Enrico, I've been stealing people. So both Mimo and Francesco came from Enrico's lab, and I think you'll be able to tell some of the tech transfer that happened. As always, it's really when a person moves between sites that you really get the best tech transfer. So I'm now going to try to give you a feeling for what we normally are trying to do, which is to sort of go in and watch embodied cells. How is it that a cell executes a developmental program? And we've come to use, instead of cultured cells or instead of spheroids for most of our work, we try to use embryos because embryos are relatively small, relatively robust, and so we can image them on the stage of a microscope, and they're small enough actually to fit there. And that, to be able to watch the cells, this is about a 21-hour-old zebrafish, if I remember right. To be able to watch those individual cells for a day or two, as we start to do that, what we see is that the cells actually have a lot of different behaviors. We could curse at that variance, but because we're trying to understand the systems biology of how those cells are interacting, what we really are now trying to do is to embrace the dynamics, embrace the variance, and use that as the signal. And that's sort of, it's not noise, it's signal. In Enrico's case, it's not background autofluorescence, it's signal that you can deconvolve. In our case, it's looking also at the expression levels and their covariances. And so we've been building multiplex and multimodal tools to try to do that. And as I say, most of the time, our techniques are motivated by people coming in or people within the lab that have a problem that doesn't quite click. And this is one of our easiest ones, and that is that there are people in the lab that are using omic scale, genome scale technologies for looking at gene expression. This is a paper from another lab and yet another paper from a different lab, and others that are looking at gene expression one at a time and spatially. And the big problem is that they're very, very different. What we'd like to do is go over to spatial genomics and really ask how much and where different messages are expressed. But the technologies that are typically used are very nonlinear. If we want to see where an RNA is expressed, then typically an antisense probe is used. And if they just was tagged with a dye, there's appreciable autofluorescence in the tissue that's hard to separate out from the signal you'd get from the binding of the dye to the RNA in the cell. And so typically, instead of using a dye, an enzyme of some sort is used, and that deposits a chromogen, as shown here. It's a microprecipitate that's being formed. And the problem with this is always that it's very nonlinear. There's a relatively narrow region at which, as things get brighter, you can deduce what the level might have been. But for low amounts of gene expression or for very brief times of staining, there's no signal at all. At long times, it saturates, and then there's some sigmoid or higher order function that links the two. And so this is actually a friend's paper that I won't cite, because it's the same gene at the same stage in the same strain of mouse, and in the same paper. And I don't think any of us could vote whether or not those two patterns are the same, or make a quantitative assessment of how much of the RNA is there. So we decided to embrace this and take advantage of some of the RNA engineering that was being done at Caltech at the time, the sort of DNA origami and other people in the department there, that in particular Niles Pierce, and he and Dirks were developing a technique that looked at the programmed assembly of hairpins. In this case, we took those hairpins and put dyes on them. And these hairpins all have a little bit of a toe hold that's complementary to the other hairpin, and a toe hold that's complementary to the other. And because the hairpins, if my finger and thumb were the hairpins and my fingertip is that toe hold, energetically they'd really rather be like this. But to do that, they would have to open up, sacrifice all their base pairing, and find one another in solution when they're at almost infinite concentration because they're a hairpin. So kinetically, if it did open up, it would close back again and stay a hairpin. So as a result, these hairpins stay very stable. If you add even blender extract of an experimental animal and run them on a gel, then the hairpins all run as this small molecular weight species. In this case, since they depend on the chemical, the sequence, they end up being chemically orthogonal. So I could take 4 pairs of them and individually use them to amplify. What causes them to amplify is the presence of an initiator. It has a sequence that's complementary to the toe hold, and then it can open up this hairpin because it has the complementary sequence. And so a branch migration takes place, which ends up with this longer duplex and a little floppy region, which is complementary to this toe hold and that. So what you get is explosive growth whenever the initiator is present. And here with these 4 different colored hairpins, if I run them all together, they run as a small species. If I add the initiator for the blue hairpins, then they appear as this high 200 mer. If I add the green hairpin, it appears as this high molecular weight species. And so what this gives us then is a triggered amplification. The triggered amplification can be made into an in situ hybridization technique by using an antisense probe and some of the initiator sequences. So when I add the hairpins, then they amplify, but these dyes stay on a leash. When we measure how quantitative it is, it's good to about 5%, maybe a little bit better. Unlike normal in situs, you don't have to critically time how long you've developed it, and it's stable in the fridge. This one spent 60 days in the refrigerator. Long Kai at Caltech is now doing 100-plex. In fact, I think 249-plex or something is what his latest accomplishment showed, using this technique, staining serially and then re-identifying the same dots of the mRNA. So this technique that we call hybridization chain reaction is similar to some of the other techniques. You can use direct labeled probes, but for thick things and for high background, they fail. The branch DNA is not, amplifies, but isn't as quantitative. And so we've been having fun using it, and more recently, building upon that technology to make something that's a little bit faster and a little bit brighter that I don't have enough time to talk about today. But if anybody's curious, write to me and we can help set you up with some reagents. Long Kai is now going over to some of those. Now, we're limited by the chemistry, and we think we've got a solution to that, and that seems to be working well. The other thing we're limited by is the way that the fluorescent dyes look, especially in complex environments like tissue. So in this case, there's two different fluorescent proteins, green fluorescent protein and citrine. If we make an experimental animal where all the blood vessels express citrine, or in this case, all express GFP, all the muscle cells express citrine, if you look at it by eye, it's pretty unremarkable. You can't see the difference because of the huge overlap. But using, and you can see that if you tried to cut between the two with filters, you might be able to get the GFP only, and that's pretty good. But if you try to get the YFP or the citrine only, you can see that the two bleed through on one another. So over the last few years, my colleagues and I have been playing around using some of the technologies that were developed first for the remote sensing crowd. If we have these overlapping emission peaks, the idea would be to take, for each pixel, the spectrum, not unlike Enrico taking the lifetime for each pixel. And so what we end up with is an image stack, X, Y, and lambda, using a multi-window PMT, for example, or other approaches to separate out the different colors of the light, and then using an approach, an inverse problem, called linear unmixing to try to separate them. And when it works very nicely. Here's individual spectral bands, only 8 of them in this case, taking 2 different green dyes, GFP and FITC. Their spectra overlap pretty dramatically, but we can take this set of images and that set of basis spectra and solve the inverse problem and say which of the signal in each one of those pixels had to have come from the FITC phalloidin, so the actin, and which of it was from the histone. The good news is that it works. The bad news is that it doesn't work as well as it should. And the problem is that an inverse problem takes time, and noise always propagates through inverse problems. So the square root of n noise, the unavoidable noise as there's collected, say, in the green image, gets imposed over into the red image because I have to try to separate the two out by solving this inverse problem. And it might be visible to you there in the audience. It looks like there's a little bit more noise in the red here, and it's not because it's really a variance in the red. It was variance in the green that got imposed. This limited the adoption of these approaches and limited our ability to do, say, a dozen or 2 dozen labels at a time. And that's where Enrico and the Fraser approach really has helped us. So taking the Fourier transform and taking it of a spectrum allows us to take something like GFP and citrine and separate them out on the Fraser plot. And just like with the lifetime, the sort of simple arithmetic manipulations works. It turns this inverse problem into a forward problem. If the signal is halfway in between, then it's a 50-50 mix of the green and red one. Or if it was 70-30, it'd be over here, or 30-70, it'd be over there. So it's dramatically faster and performs dramatically better. So if we take trying to separate it out by eye, it fails. If we try to separate out the muscle from the blood vessels, you can see these regions where we're getting what looks like signal, but can't be because there's no blood vessels there. And if we use the hyperspectral Fraser approach, then we get both a dramatic speed and speeding up of the processing, and we also lose a lot of that background signal. This has let us start to look at live animals with multiple labels and follow over time, for example, 3 different fluorescent proteins, some autofluorescent signal, in this case it's a structural autofluorescence, a chemical autofluorescence related to the metabolism of the cell, yet other labels and other autofluorescences. So this allows us to separate out a variety of things, in this case from a living animal. The magenta here is similar to some of the metabolic signal that Enrico and Andrew were showing you. There's other things that are more structural. And so because we can see this in autofluorescence from the color, it makes you wonder if you could image metabolism and other things without FLIM. And in fact, you can sort of find some of the labels, like NADH bound and free can be found in the hyperspectral Fraser, but it's not as well positioned and not as good of a solution as doing the lifetime imaging. You can see here molecules that some of us know and love, like retinol, show up, folic acid, the NADH free and bound, all of these shown in experimental embryos. But given that we've got those spectral signals, we'd like to use them. Enrico's shown you how powerful the lifetime imaging is. And what Mimo and Francesco have been now doing is to combine both of them and try to make SpectraFlim sort of things, almost a belts and suspenders sort of approach to deconvolving the different signals. So we've gone away from just using the spectral. We now are working with some of the technology that Enrico first started and add to it lifetime and hyperspectral, and then feeding it down through a bronchoscope or an endoscope. We're just starting to get our protocols in place to use this in patients, but have been doing experimental animal work so far. So that's what we want to be doing. Right now, this is rodent trachea that you're looking at. You can see through from the airway surface down to the collagen structure below. As Enrico pointed out earlier, the collagen has characteristic autofluorescence. So you can see that nicely. And you can see that if you go over into the lifetime mode, you can see the individual cells and their metabolic state as well. Enrico really painted the way for us to follow in this. What we're trying to do is now carry it into settings that aren't very convenient for microscopes, but are convenient for being able to see, for example, the effects of environmental exposure or other pathologies on airway and in other settings. So I started by saying some of our stuff gets motivated from outside. What I wanted to do is spend a few minutes on a technology that we've been involved in building and testing and finally have it through the Chinese FDA that started with Dan Swartz complaining to us on one of his visits to Caltech, I think because he wanted to have an emptier waiting room, that most of the people that are in there don't need to be, and most of the people that should be there aren't there yet until they have a big scotoma in their vision. And so what he was curious about is, could we come up with a way to see the vasculature so you might be able to see the earliest signs of diabetic retinopathy or macular degeneration in an easy way. And of course, we were very cooperative with him because we were used to imaging live beating hearts and live vasculature and all. And we said, if you just get GFP expressed in your patients, that it'll be a cinch. He seemed reluctant to go that way. And so what we've been working on is a way to try to get the sort of resolution we can get out of the fluorescent proteins that let us watch inside the animal's chest as a heart develops and differentiates, and be able to do that in the living animal, in this case, the living patient. I mean, most of the people here will know that to get the contrast that you need for an angiogram, concentrated indocyanine green or fluorescein, as shown in this picture, is injected in. A bright blue light and a flash photograph is taken through a dilated pupil. And while that works, it takes time, and it's hard to imagine people self-administering it or screening in a simple way. And so since at that point we were building OCTs and trying to look for different contrast mechanisms that we could use within the intrinsic tissue, what we started doing is thinking about what happens in that OCT as we split the signal that's coming out of the source, bounce it off of a mirror and off of the patient's eye, bring it back together, collect it down to a spectrometer, and now we get our friend the Fourier transform. The spectrometer, if we convert it with a Fourier transform, we can get a A scan. If we repeat this on many different places, you get the whole B scan. That gives us those beautiful images. And what we decided to do in our embryo work and then in the patient work was to, rather than square the signal that's coming out, which throws away the imaginary part of the signal, that is the phase of the signal, we decided we might be able to exploit that phase signal to be able to see the vasculature. And what pointed the way in that is, again, some work here from UC Irvine. So Doppler OCT, if there's blood cells moving towards the light source, should give you a Doppler shift in the signal. So what the folks here at UC Irvine were able to do is to see a blood vessel that's somewhat transverse but aligned with the light source. And so that shows it could happen, but most of the vessels we want to see are at 90 degrees and things move slow and so on. So we were hoping we might be able to come up with a different approach. And the approach we decided, and I should mention why we went for phase rather than intensity, it's easy for us to measure phase in 1 part per million. It's extremely hard to measure intensity at better than 1%, so 1 in 100. So what we are hoping for is, as things moved in the vasculature, like the scatters, the blood cells, they're going to be at slightly different distances, and if we could build an instrument that was phase stable enough, we should be able to see the nanometer scale differences in how far those are away from the observer up that way, and be able to see that in the phase variance. So what we started doing was acquiring a few different phase and intensity images, align those and compensate for the bulk motion of the subject. So you don't want to be subject to how stable their chin was on the chin rest. And then be able to take then a local variance calculation that we could combine. And the first images that we started taking took about 4 seconds. Gave us good resolution. This is one where we're starting to take more pixels and a larger field of view. The color is encoding the depth into the specimen. This is, let's see, that's postdoctoral retinal circulation, but it works on non-postdocs, although they are awfully motivated subjects. And the nice thing is that we could then compare it to, on that same subject, a fluorescein angiogram. And I think we get the nice depth discrimination that we don't get from the fluorescein. We get it in seconds in a non-dilated pupil, in this case. And of course, dilated and longer time to do the the collection by the fluorescein angiography. We've now been looking both here and in China at different eye diseases. This is some of the early work done together with UCSF, but taking a fluorescein angiogram and comparing it, we get, of course, depth discrimination, and you can see the ability to see vessels just as they're pinching off, see the microaneurysms, and also be able to see their depth into the retina. So where we are right now is that we think we've got some working suites of techniques. As Andrew and Enrico were sort of mentioning, our hope is to bring all these together into a single imaging suite where we're using the Fourier transform in many different ways to do hyperspectral phaser, to do lifetime imaging, and then either our phase variance OCT in cases where the phase stability of the instrument can be assured, or using intensity fluctuation approaches like some of the commercial instruments that are out now for OCT angiography. All of these give us relatively fast and easy image processing. We're able now, and are having a lot of fun, looking at heterogeneity within biopsies as well as within spheroids. If we can keep the tissue alive over time, we can see how that heterogeneity evolves with time in the spheroids or in the live embryos or in patients. And the spectral film now, although I've left a T out, so anyway, the SpectraFlim we are really enjoying for use in this setting. It's really taking both of the modalities that Enrico has instructed us about. We're trying to convert the background into signal, and we really would like to be able to look in at the metabolites and the metabolism of the cell, and we're very optimistic that it's going to work. So I'll just stop by thanking the people that have funded us for some of that. We've got some patents and some corporate involvement trying to make these processing and imaging techniques a reality that could be deployed out, and to thank the people that made it possible. The OCT was really pushed forward by Jeff. A lot of the imaging in the lab with the HCR and other things was pushed forward by VCOS together with Ty and many others in the lab. And of course, Mimo and Francesco have been mainstays in our continued stealing from Enrico. And I will stop there and see if there are any questions. So we haven't tried the photoacoustic for a few years now. When we were trying it, we were having problems getting the resolution that others are achieving now. And I think it would be worth reattempting and seeing even if we could do spectroscopy versions of photoacoustic by modulating the light, either chirping it or in other ways changing the intensity. Right, so could we use the autofluorescence techniques that you've developed to look at the metabolic state of the cells? Could we use the OCTA to look at the vascular tree? And could we use photoacoustic to tell us about the local oxygenation or other things? I'm optimistic that we could. I'm not optimistic that we could do it quickly. And so what we've been trying to push on in this project is to do it fast enough that it could be used in screening as a tool that might take tens of seconds in a non-dilated pupil. What we're hoping is to eventually make a research instrument that pulls all those modalities together and even also probes the physiology of the photoreceptors, because the other thing I think that would be worth bringing in would be to answer the questions about the physiology of the photoreceptors, both looking at the amount of rhodopsin that's there rather than the reflectivity of the outer segments, and to look at the recovery after a partial bleach. We've done some of that imaging in the past, mostly in rodents, a little bit in people. Again, it's hard to do it quickly, and the people have to sit in red goggles or sit in the dark for a while. My students are used to sitting in the dark, so there are subjects around that can do it. But what we're very interested in is, can we make it fast enough to make it practical as a screening tool? And then the second instrument we'd like to build would be the research sort of tool that I'm sure you're imagining, that would let us pull all these different metabolic and physiologic signals together. Yeah, I'm not sure if I have any of those, but it works nicely for that, and we've been collecting some images now where we are unmixing it. An example of that is shown here. None of the cells are completely oxidative or completely glycolytic, so we can see shades of gray. But what we've been doing in a lot of these, and you might even be able to see it here if we blew it up a little, there are many of the cells that are multiply labeled. And some of them where the labels are co-distributed. This is not in situ, that's living, and it's fast enough. And so this is so what we've been trying to do is come up with techniques that could be applied both to biopsies in a fixed state by adding, say, the in situ hybridization probes I was mentioning. That has been quantitative, and we've been able to separate out dozens, and as I mentioned, long Chi's up to into the hundreds of different genes that are co-expressed on a cell-by-cell basis. Then we're trying to deduce what the gene regulatory network is that explains that co-expression. Or in the case of fluorescently labeled cells like this, we want to understand which reporters are either of gene expression or of cell biological phenomena are co-distributed or co-expressed. And in this case, it's multidimensional because there's multiple labels, plus we're following it over time. So it's, and then trying to unmix it, I think, as you're suggesting.
Okay.
Does that make sense?
A bit. The technical aspects are beyond me, unfortunately.
Well, I mean, these are all live, and no one cell has only retinol or only retinoic acid or only NADH. So the goal is to be able to separate those out from one another. And I can show you some examples once I'm not on the stage here of cases where there's multiple things expressed in the same cell where we're trying to separate it out. And what makes it possible in that is that as you take an admixture of the two things, if it's 50-50 in the same pixel, then it's halfway in between. If it's not 50-50, it's just 50-50, but they're neighboring pixels, one pixel would be over here, the other pixel would be over here. So the real power of the spectral phaser is graphically, you can see very quickly on a pixel by pixel, or if we segregate the image, segment it based on a cell by cell, you can see if the cell expresses only red, only green, or some admixture of the two. Does that make sense? And I can show you a couple things where we've analyzed both cases where there are nearby protein labels or where they're labeling the same complex. It's in the same diffraction limited spot. Okay, well, thank you very much.
Thank you.